Hi everyone, welcome to our third IMMUNO-model webinar. And today we will listen to the talks and discuss the field of mathematical oncology. And I would like to welcome all the speakers. And firstly, I would like to give a floor to Professor Peter Rashkov from Institute of Mathematics and Informatics from Sofia, Bulgaria. So he can introduce the speakers and then we can start with today's talk. So Peter, the floor is yours.
Thank you, Barbara. Welcome to this webinar where we will speak about a little bit different topic compared to the other webinars. Namely, we'll talk about applications of mathematical models to oncology. And I have invited two speakers that will present different models. Both of them have worked extensively in the field of mathematical modeling to different biological processes. And I hope that you enjoy the presentations. After the two presentations, there will be time for questions. So please reserve your questions for the end of the webinar. And with this, I would like to present the first speaker. This is Professor Christina Kuttler from the Technical University of Munich, where she's an extraordinary professor in the field mathematics in life sciences. She did a PhD in mathematics at the University of Tübingen in Germany. And afterwards, she continued as a postdoc there, where she worked on mathematical modeling of granular matter and prediction of proteasomal cleavages in the context of the immune system. Afterwards, she got another postdoc at the Institute of Biomathematics and Biometry at the Hinow Center in Munich, Germany, where she worked on various problems in biology from mathematical perspective. And her research is focused on different questions such as models of bacterial communication, intracellular regulation, and diffusion processes, and also applications to mathematical oncology. So I would like to welcome you. Please, if you could start your presentation.
Thanks a lot for the kind introduction and thanks for organizing the webinar. So I'm really looking forward and hope that I can show you a little bit of the results we have worked on and also introduce a bit in the way of thinking we may use for this. Okay, so I would like to talk a bit about how we could use such mathematical models also to predict tumor dynamics and metastasis. But before we get to the details, I would like to give you a quick overview. So I would like to give you a very brief introduction into the modeling. Don't worry, there might appear some mathematical equations. I don't expect that we get into all the details, but I would like you to see them at least and to understand a bit about how the structure is, as I think that helps also to understand how we can use it and apply it then. And mainly I would like to focus on two case studies that are projects which have run with a PhD student of mine, Pirmin Schlicke, and show you a bit what we could do there with the models and how these models look like. Okay, then let's jump into maybe first of all to speak a little bit about why should we use mathematical models. So of course, mathematics is not so easy always to handle. So is there any advantage of in general? And of course, I would say from my perspective, yes, of course there is. For example, when we try to model tumor growth, it may help to understand the dynamics which is behind. So maybe we can understand the processes, or we can set up something like hypothesis, what might be the main influences or so, for example, on how cancer cells proliferate or so. And what I would think is one of the main advantages, if we understand these processes and these dynamics and mechanisms and so on, we may be able also to predict future behavior. And of course, that's something where we really need the models, the mathematical models. One could think about, for example, then also how to optimize treatment strategies. Also there, we need to understand the principles behind, and then with the mathematical model, we can play. So we do not hurt any patients or so, but we can try what would happen if we do the treatment differently, maybe different time points or have different combinations or so. And these mathematical models would then also allow, if corresponding data are available, that we could design personalized treatment plans. For example, if one learns how a cancer disease grows in a patient, we may have ideas more concretely there. Of course, what such mathematical models also allow is to analyze data. So, for example, to find something which is not directly visible from the data, but which one can elaborate by that, something like trends or correlations or so. And of course, one, from my point of view, also important thing could be trying to understand the interaction between the tumors and their environment. So what's going on there with chemical signals and so on, immune responses, and many more things. Okay, then let's jump into. Let's start with a very brief overview. I guess maybe you have seen something 2 weeks ago about some simple growth models for tumor cells, but also in general, just to give you a feeling. So typical description for such processes is in a mathematical context to use so-called ordinary differential equations. And as you can see here, this dot here represents a derivative. Derivative has to do something with changes. So n describes the number of tumor cells at a time point t. So the derivative says, how does this number of tumor cells change in time? And the classical logistic growth says something like, if there are only few cells, it's growing more or less exponentially, but then there is some limitation, and the limitation is formulated in terms of a capacity denoted here by K. That would be a very classical approach in the context of tumor growth dynamics. One often also uses, instead of the logistic growth, the so-called Gompertz growth. That's quite similar. Also there is a K in which gives something like an upper limit for the number of tumor cells which can be reached. As obviously in a patient's body there are limitations, so it cannot grow to infinity or so. Okay, that's very basic, of course, and I would say maybe oversimplifying, not helping too much, but a little bit at least. For example, about growth rates, which is covered here by this parameter denoted by R, that would tell how fast the tumor would grow. Okay, but of course we would be interested in understanding more, and one thing could be that we want to include therapies into the modeling. We want to understand how they act, and some of the therapies may act better or mainly, for example, on proliferating cells, but not so much on quiescent cells or cells which are not just dividing, or even dead cells, of course. No result from the therapy. We may want to take this into account, and what you can see over here in this sketch of a structure are different types of cells. So one would distinguish this into so-called compartments, having the proliferating cells, having something like quiescent cells. So I would say the cells which are not proliferating at the moment, maybe also dead cells. And then we have transitions between these states, as you can see here by this structure. And if we take this to the differential equation system, we get now 3 differential equations for the 3 compartments, again with the derivatives. That means how do, for example, number of proliferating cells change. Have again such a structure which takes into account who enters the compartment of the proliferating cells, who leaves that, and so on. And so we get such a to such a system. That's already a bit better. Nevertheless, it's still a bit simple in the sense of we just consider the numbers and not something like which allows to take into account a spatial structure, for example. But of course, if one thinks about a solid tumor. Of course, it may play a role where exactly it's located, what's the shape of it. Maybe there is even more spatial heterogeneity inside, how is the tumor arranged in a spatial situation. And for that, we can get to more structured models, which take into account such things. And then we may end up with larger models. So what you see here in this little sketch is, for example, we have still the cell division so the cell growth, but maybe there's also spread in space. So some cells may just move. Maybe some want to follow chemotaxis. That means follow a certain gradients of chemical signals or so. But of course, also by growing, they need some more space and may spread a bit. And what we see here is, again, a quite simple model. On the left-hand side, you again see a derivative. What we need now is mathematically a so-called partial derivative, as our cell density may depend now on time as before, but also on where exactly we are. And so we have two independent variables, the space and the time. And so we need so-called partial derivatives. Here again, how does it change in time on the left-hand side? And then on the right-hand side, we can combine different effects we want to include. So for example, this first term describes diffusion. So as you imagine diffusion, something which is an undirected movement of the cells, just to get to areas where there is a lower cell density, for example. Then we have here the second term. This would cover chemotaxis. That means following maybe something like going towards a higher concentration of some substance. Haptotaxis, another possibility how to move. So we can extend it. So it's like a collection of different effects, and we could make assumptions how they work and then put it together in a mathematical equation. That's roughly the procedure. And what you see already here, there are a lot of parameters included here in the general setting, symbolized by these letters. And practically, for a concrete patient, of course, one needs values for these parameters, and that will cause some problems. I will get to that later again. Okay, so what are typical problems? What can we expect from these models? As said before, we need concrete values if you want to say something quantitatively at least, but often it's impossible to find data which are adequate or which can help here in this context. And one typical problem is one would like to start with a model without treatment at the beginning to understand the principles. So how would the growth and spread and so on work if there's no treatment applied on the patient? That's quite difficult, as of course, typically patients are treated. Sometimes one can help a bit with mouse models also, but sometimes it's also difficult and one needs to guess or so. Then thinking about practical life, of course, from clinical routine, there are data points available. So for example, one does a CT and then knows maybe how large a tumor is, but you do not do that every day or every week or so, of course. That means only a few data points may be available. Sometimes it's that you cannot directly measure the quantity you would be interested in, but only indirect by, for example, looking how is the growth, but you do not know how are some molecules distributed or so. That's a problem. Another problem is, of course, there's a lot of patient specificity in, as different patients may have different mutations in their tumors. The location of the tumor may play a role. Of course, what's the general health condition of the patient, and so on and so on. That means it's quite difficult to find valid data, but in principle, we would need them for being able to make quantitative predictions. And that also means if we set up such models, we should keep that into account, that we do not blow up the models to incredibly large objects, and we do not have an idea how to fill the values in for the parameters which are included. It also means it's more than just extrapolating. With this cartoon, I wanted to express this as these models are highly nonlinear and, of course, interacting mechanisms behind. So we need to be careful with that. Okay, now let's jump to the first case study. I would like to give you a brief overview about that. So that's a very concrete situation. We wanted to focus there on non-small cell lung cancer and compare the model outcome with observations from 2 patients, and these patients both received immunotherapy. So what you realize, you may say, okay, 2 patients, that's really not a lot. Yes, that's true. But that was how we could get some quite nice data, at least for our purpose, to know about tumor sizes, and had some time course for that. So the goal would be to catch maybe the most important effects, such that we can reproduce the data as a first step. Also to take into account how these patients were treated concretely. So the time points or time periods are known when they got treatment. And finally, we can use the model maybe to test just in the computer with the mathematical model what would have happened if we have alternative treatment schemes or so. And in principle, what we have here is a first step to enable clinicians also to make informed decisions, which means not do something like try and error with patients or so, but first try with the model what would happen, what maybe would make sense, and so on. Okay, then let's jump into. So we want to simulate the tumor growth, cancer growth. We want to take here into account really a spatial model. That means exactly where is the tumor located, how does it look like as a volume. We represent the tumor in the model by so-called volume fraction field. That means we give values to each point in the space, and the values are between 0 and 1. So 0 means there is no tumor. 1 means it fills completely the volume with tumor cells, and in between there can be something with empty space or with other cells or so on. We will use again such a partial differential equation system, describing time course and taking into account there the dynamics. And for the final outcome, we would like to see, can we cover what happened to the patients in response to their therapies. A first step is an important one and a quite time-consuming one. One needs to construct, in this example, the concrete patient-specific lung geometry. That's what you can see here on the left-hand side. There's also included how the blood vessels are located. That is quite a lot of work to get this. My PhD student did this very thoroughly, extract from the corresponding CT images. There's so-called 3D slicer used. That's an algorithm tool. What you see on the right-hand side is this blue slice you can see over here. And you see here a kind of grid that's called a mesh. That's something we need then for the mathematics to let run our algorithm for the for describing the time course. You see that's 2D. That's something one often does in mathematics, if possible, trying to reduce as, of course, a 3D object that's quite large. And if we want to describe there many points in a 3D setting, that can be quite tricky and time-consuming also for good computers. Okay, then here is the model. Don't worry, we don't need all the details. I just wanted to give you a very brief overview. So again, we have here on the left-hand side something which describes how does it change in time. And here we consider the proliferating cells. How do they change in time? And their fraction in the volume. So we have one component which describes how they spread in space. We have one component which describes how they grow or how they are degraded. And what goes in here is what is denoted here by the phi tau. That includes the immunotherapy. So how does it act on the cells? That's covered here in a bit more detail. So here we have this reaction term. So we have here in the reaction term for the growth, we take a modified version of the Gompertz growth. So a very simple one, just a bit adapted here to the peculiarities here. Then we have the effect of the immune immunotherapy as a second term, and a third term was important here. So if there's just nutrient lacking, then of course also the cells could degrade or vanish or so. We also need an equation which describes the level of the immunotherapy. Also that changes in time. So we may have some drug influx, that's the second term here, which takes into account when does the patient receive the therapy. That's here just notated in the sense of when is the therapy given, and then this is relevant. There is then the influx, maybe by infusion or so. And the first term, that's including just the decay, so what's the half-life time, and by that it's degraded and vanishing. There's also nutrient distribution included. I don't go into that into detail, as then the model gets even larger. Just to mention, it may be helpful at even not to go to 2D, but to 1D, by assuming that the tumor might be radially symmetric. Of course, that's not completely the case. Nevertheless, it may help to understand a bit better, and the calculations for that is much faster, and by that may make sense. Okay. So we have the model approach there. Of course, there's a lot of development behind, but now let's jump into how to calibrate the parameters for the individual patients. One problem is maybe not the whole tumor volume is visible. We need to distinguish what is visible, what is invisible. Of course, the model does not directly distinguish between that, so we need to take this into account. Where are maybe only few cells you couldn't see in a CT or so, may play a role. Then we can directly jump over to one patient. So we see here time course from left to the right. That's in days. Maybe let's start with the upper figure that covers the tumor volume, and you see these crosses. The crosses are the measurements for the tumor volume. So there are quite some available, but the time course was also quite long. You see here these marks. The E was there the patient had an emboli. So of course that was a problem. Then at P there was a therapy break. And finally, he received also dexamethasone as an additional treatment. Okay, that's the tumor volume, and you see it initially it's increasing quite a lot. And then after some time, that's the second figure, the doctor started with the immunotherapy. We see here the calculated level of the immunotherapy acting until here, P, where the therapy was stopped. And then, of course, the level goes down again. So what we have here is just pharmacokinetics behind. And here we see the mass of the tumor cells. So that's also including invisible parts thereof. And what is maybe also visible up here, so the blue one here, you may see here a little bit, that's what the model tells us, so how we could fit the model to the data, and that fits quite well. Okay, so this was one patient where the therapy kept some control, at least for some time. Here we have a second patient. There it did not work as well. Here I have to say the therapy was already ongoing when the measurements we had were started. After some time, the administration schedule of the drug was changed from 3 weekly to 6 weekly. And finally, here in this context, unfortunately, the patient died. So there was PD, that's therapy end, and exitus letalis finally. Okay, also these things can happen, of course, for such a severe disease. What we can see here in the lower one is that the tumor mass, what we consider here, is growing despite the therapy. That's something which is not directly visible here in the data. So one could have the idea, okay, here it's under control. Unfortunately, it's not. The model realizes this, that there is some problem going on. And to show you quickly, about what can we do beyond observation and just reproducing what was observed in the data already. We could, for example, test what would have happened if we treated the patients with different therapy plans, for example, give them lower drug dosage or so. And what you see is here for the first patient, then the levels of the tumor volume would have risen earlier. And that's not surprising, but we can make here even a kind of quantitative predictions for that. Similar for the second patient, of course, people may ask, would it have been better to give him a more aggressive therapy or so? Unfortunately, what we can take out from here, it may have delayed a bit, but not too much. It's more about the time point, but here in this situation, it wouldn't really have helped a lot. And just to show you one figure, one could also go beyond the observation in the sense of we could make spatial simulations, so really try to predict how would the tumor arrangement look like in the time course, how large is it at which time point. Okay, maybe very quickly, as I'm running out of time, we can also consider not just the tumor growth, but also what happens with metastasis formation. Maybe let me jump over this and tell you a little bit more about what we can do there. So also there, we would be interested, when do metastases appear? What are the typical problems we have here? Again, the different patients with different therapies, other health issues. A big problem for metastasis is, of course, if they start to grow, they are tiny, they are too tiny to be visible in a CT or so. But maybe the model can help to predict when do we expect metastasis to appear, when we have learned a bit how is the tumor type of this patient growing. And indeed, that's something which is possible to do. What we see here is indeed tumor dynamics under treatment. So in these first two figures, we see here on the left-hand side just the total tumor burden. That means how much tumor mass is in this patient in total. The gray area is when the patient received some therapy, shortly chemotherapy, later for longer time immunotherapy. And you see the data points which are going down. The model does the same. So this curve represents how the model could be fitted to this data. In comparison, if no treatment was given, then the tumor growth would have been expected like this curve here. So obviously the therapy here makes a big difference. Right-hand side is just the drug concentration for the immunotherapy in time course. And maybe even more interesting is the metastasis density distribution. What we see here on this axis is size. Have a look, that's in orders of magnitude. We only can see metastasis in the CT if they are in this upper area in the right-hand side. The red ones here that are indeed metastasis which have been observed. And these blue diamond points, they represent what the model gave as output. And one can also here fit quite well. Same on the right-hand side, that's just later. And what you see, there was the therapy going on. The metastases have become smaller. And also to hint on, maybe such simulations could hint on some metastases which are yet invisible in the CT, but already present. And by that could help the medical doctors to decide when maybe to check in CT or when maybe to start with the next therapy, or also to avoid that metastases grow. Okay, let me sum up. So I hope I could show you a little bit that these mathematical approaches can help us to understand the tumor dynamics. To some extent, predictions are possible. Nevertheless, we rely heavily on data as far as they are available in clinical routine. And on that also the quality of these predictions depends strongly. Then I thank you very much for your attention and looking forward to questions afterwards, after the second talk. Thank you very much.
Thank you very much for for this wonderful presentation. And now let us go to the second speaker. This is Professor Peter Hinow, who is originally from Germany, but he is a professor now at the University of Wisconsin-Milwaukee. And he also works in mathematical modeling across different areas, such as mechanics, biology, ecology, tumor, host interactions, drug delivery, etc. I would like to invite him now to give his presentation. Thank you, and the floor is yours.
Okay, thank you. Can everybody hear me reasonably well? Okay, very good. So very nice talk, Christina, and she did a lot of work to make my talk much easier now. So I'm going to talk about a mathematical model of the disruption of glucose homeostasis in cancer patients. And so first of all, I want to acknowledge my collaborators on this work. The primary driver behind all my work on mathematical modeling of cancer is Emily Wang, with whom I have been working for over 20 years now. It's amazing. And she's a cancer biologist, and she works in the Department of Pathology at UC San Diego. And her work is actually very classical in the sense that it produces rarely ever any numbers. I mean, we have one paper or maybe two together where we had some cell numbers of cells that were grown in dishes and subjected to some treatment. That's actually the exception. Most of the work that she does is on siRNA and vesicles and stuff like that. So it's actually more of a informatics type of data. So yes and no, something lights up in red or blue or green. So that it's really unusual that she has some actual experimental data that mathematician could easily use, especially if you are working with differential equations. So this paper that I'm going to present actually didn't really contain any data in it. We're going to see a little bit later at the end what we use to justify our modeling work. And then also we've worked with Gabriela Pinter, who's a colleague of mine in the same department. And we also had several very talented undergraduate students in mathematical sciences who helped us at different times of this project. So first I want to talk a little bit about the altered glucose metabolism and so-called insulin resistance that is often observed in cancer patients. And then we're going to show you how we built the mathematical model. So it consists only of ordinary differential equations, which we already saw in Christina's talk. And then I'll say something about the parameterization and simulation, and also how does this model and its prediction match what has been observed in the clinical literature. And I'll finish up with some future work or possible topics for future work. Yeah, so cancer is, among all the other things that it is, also an energy burden. So the cancer cells, they are rapidly proliferating, and they need a lot of energy to build up their cells and to proliferate and to grow. And what they do is they basically gobble up all the glucose that is available for their own use. And on top of that, they are also preventing the healthy cells in the body from taking in glucose. And so this is a situation which is very closely comparable to that in a diabetic patient. In a diabetic patient, the cell's uptake of glucose is also impaired. And then it's a observation that in let's say Western industrialized societies, a lot of cancer patients already have certain compromised health. In particular, they can come into the cancer disease with diabetes already present. So their glucose insulin metabolism is already disturbed. And so now on top of that, the cancer makes this aspect even worse. And over the years, there have been several proposals to use anti-diabetic drugs in another capacity, namely to support the treatment of cancer patients. So this is not a kind of treatment that is usually associated with cancer chemotherapy, where the goal is to kill the cancer cells. Instead, here the idea is that one could reduce the burden of the cancer on the general health of the patient, instead of trying to necessarily cure it. Yeah, so here is a schematic of our model of insulin resistance. So we have just 3 variables. I intentionally kept the number of variables small, so we're not even considering insulin separately. So we have the population of the cancer cells, that's denoted by B of T. We have the glucose-dependent healthy organs, so basically everything but the skeleton. And then we have the glucose reservoir, which we assume to be the amount of glucose that is permanently circulating in the blood. And so these arrows here are the various interactions between those populations and the glucose reservoir. First of all, we all have to eat. We need to continuously replenish our glucose reservoir. And then both the cancer cells and the healthy cells, they consume that glucose, but the cancer is able to suppress the uptake of the glucose by the healthy cells. And so in a way, there will be more of that left for the cancer to consume. Yeah, so from such a picture, it's fairly easy, fairly straightforward to build an ordinary differential equation model. So it's almost like this picture is encoding the set of equations that you're going to see in the following slides. So here we have our variables, the amount of glucose in the blood, the glucose-dependent body mass, and the mass of the cancer tissue. This is already a substantial simplification. So we are really only looking at the blood glucose. And of course, there are other reservoirs for glucose, such as the liver. But here we're just looking at the amount that's continuously available in the blood. And so first, of course, we have to start with a cancer-free case, so where there are no cancer cells, and we have only 2 variables, which are the glucose and the body mass. And so here we have the supply through the food intake. We have this consumption of the glucose by the healthy body, and we have excretion, which normally is small. So there's normally very little loss of glucose through the urine, which, however, can be higher in diabetic patients. And then we have a loss term here in the equation for the healthy cells, but this is just to make to create a steady state, right? So normally, if you live your healthy life, then you are at steady state. Your body mass is not supposed to change significantly during a cancer-free state. And so from these equations, it's a very classical procedure. You set the left-hand side to 0, and then you solve for the so-called steady states. So those steady states are known, they are observed. So we choose that you have about 4 grams of glucose continuously circulating in your blood and a non-diabetic person, whereas the number is significantly higher, about 11 grams in a diabetic person. And the healthy body mass, of course, that differs vastly from even healthy people, but here we use 60 kilograms. And you can use other values which would not significantly change the outcome of the model. And so these 2 equations here can be considered constraints. And in fact, they are actually needed to fix some other parameter values which we were not able to obtain from the literature. So we're doing a little bit of a back on the envelope calculation. So now we introduce the cancer, and that changes the equations that we had before significantly by this term that's marked in blue here, which basically models the downregulation of the consumption of the glucose by the healthy body in the presence of the cancer. And so you see this denominator is going to make this term smaller, and which will leave more glucose behind, which then gets consumed by the growing cancer cell mass. There's also a death term in here for the cancer cells. And yeah, so I'm not going to go through a lot of this parameterization. The difficulty is that a lot of these things are not available in the literature. And so what you have to do is you have to make some guesses, and you have to also use some of these equilibrium constraints that I showed you earlier. And then eventually you have to also see if you can replicate plausible developmental histories of cancer. And I'll show you in a moment how we made that work. Yeah, so there are certain choices that we had to make. One is that the cancer cells consume glucose at a higher rate than healthy cells. And this is also known because they express certain glucose transporters in higher proportions than healthy cells would do. And now there's one thing to say that they consume glucose at a higher rate, and it's another to put a factor between those constants, right? So here, for the lack of a better, more obvious choice, we choose that the cancer cells consume glucose at twice the rate as the healthy cells. However, this can be changed, and this is also one of those advantages of a mathematical model, that one can also play with these constants and see how that changes the outcome. Yeah, so here we have a sample simulation of a non-diabetic patient, which, so I didn't say much about the initial conditions, but the initial condition of the cancer is that it was 0.5 grams. And so you see that here over time, the first of all, the cancer mass grows, and we stop the simulation once it reaches 100 grams. This is also an arbitrary number, but you just set a maximum and then you just call it a day. And at the same time, also the healthy body mass decreases. It's not much, it's maybe like half a kilogram. That it goes down until it reaches this point here when the tumor has reached 100 grams. And over time, the glucose in the blood also increases. The two curves, the difference between red and blue, is that one of these cancers is more aggressive. The time to reaching 100 grams is shorter, and the other one is less aggressive. And this time here is approximately 8 years. Yeah, so now we also have a different alternative parameter set for the diabetic case. And so here we see that in the case of the diabetic patient, this is the red curve here, the time to reach 100 grams of cancer mass is much shorter than in the non-diabetic patient. Yeah, so that allows us to define certain cancer characteristics, which are, for example, this time t star that it takes to reach 100 grams, and then also the tumor doubling time, and also the total weight loss relative to the initial weight of 60 kilograms. Yeah, so there now you can have you can compare these observations or these predictions from the model to observations in the literature, and those are vast, and you can find papers on different cancer types, different countries, pan-cancer studies, and so on. Right, so couple of possible values that I extracted here are doubling times of lung cancer, and they can come anywhere between 30 and 1,077 days. However, the mean is something like 160 days, which now we can compare to our values here. It's a bit smaller than what our model would predict, but still, I mean, it's roughly this correct order of magnitude. Right, so among breast cancers, we found there are doubling times between 150 and 250 days, which is surprisingly constant over almost a century now. And yeah, so it's again roughly what we have observed in our model. And then certain mean value of fasting glucose level in breast cancer patient is slightly elevated compared to that of healthy, I'm sorry, yeah, so for a control group, right? So the non-cancer patient. So the factor is about 1.1. It's not that much, but it's still elevated. And yeah, so again, all these observations that we can find, they mirror the predictions of our model, right? And yeah, so then we also looked at this time t star that we defined as the emergence of the cancer. And yeah, so this is generally believed to be several years to a decade. So there's not really a fixed value that you can find anywhere in the literature. But again, it's similar to those 5 to 8 years that we saw in our model for this T star. Yeah, and then diabetes and hyperglycemia are associated with a higher mortality in patients with non-small cell lung cancer. And yeah, so again, it's in line with what our model predicts. Yeah, so then the one last thing that I showed you earlier, and maybe I should go back here, is this slope of the weight loss curve at the time T star. And this can also be compared. Here we have a value that patients with stage 3 and 4 non-small cell lung cancer, they lose weight at a rate of about 60 grams per week. And that's a little bit larger than what our model predicts. But this could also be the result of a full-scale cachexia, which is basically the complete loss of skeletal muscle and adipose tissue, which is being converted into glucose to feed the cancer, right? So this is not contained in our model, but future version of it could include cachexia. Yeah, so then we also have the potential to try various types of drugs, which basically in the model affect some of the parameters in one way or another. They increase it or they decrease it. And so we can define different types of drugs, which, for example, could increase the uptake of glucose in the healthy cells, or they inhibit the reabsorption of glucose in the kidneys, or another, we call it the type R strategy, which decreases the dietary glucose intake. So in a way, decreases the supply of glucose. And so then you can play those things out and see which of these has a positive effect in the sense of increasing the time T star to outbreak. Yeah, so one of these drugs that has been approved already in a couple of cases is metformin, which has been used to treat diabetic patients for many, many years. And yeah, so it has shown some positive outcomes in certain types of cancers, but in others it hasn't shown any outcome. And that is leads to a natural question: what makes these different types of cancers different? And of course, this is at the type of cancer level. And then the next level would be, how is one patient different from another, even if they have the same type of cancer, right? So yeah, this is a bit of a shortcoming of our model. We have it simulated only this abstract type of cancer. There's no distinction between well, lung or liver or whatever else, right? And so in different types of cancers, I'm sure that these parameters have different values. And again, there's also this possibility that different patients with the same type of cancer also have different values. Right? So one that would be perhaps accessible to investigation would be this glucose consumption rate, which could be derived from PET scans with radioactively labeled glucose. Yeah, and then I was talking to a colleague at the University of Copenhagen, and so she informed me that in exercise can contribute to improved glucose metabolism and yeah, in improved glucose uptake by the healthy body mass. So they're trying to get cancer patients to do exercise, however difficult that may be, and hope that this has a positive impact on the course of the cancer disease. Yeah, and with this, I'd like to thank my collaborators, and the paper, if you're interested, is available on bioRxiv. Thank you.
Thank you very much for this talk. And now we have time for maybe a couple of questions. So please, if you would like, just put the questions in the chat, or someone is raising a hand. Okay. Okay, so please feel free to ask questions here, or maybe in the Q&A. I don't see anything.
I have a question for Christina.
Okay.
So could you show up your equations once more? Yeah. Yeah, so this spread in space, it's a gradient of mu p, right? And so then that mu p itself seems like a differential operator, and so is there another Laplace inside the that suggests like too many derivatives? Is that Laplace there?
Hey, maybe somebody is muted. I don't know.
Yeah, I think Christina is muted.
Sorry, I was speaking. Sorry. Yeah, doesn't make sense if I'm muted. Sorry. Yeah, indeed, it's a fourth order. What we have here with this Laplacian, that's correct. That's this generalized Kern-Hilliard model approach.
Oh, okay, yeah.
Yeah.
Is that? I mean, it looks a whole lot more complicated than a
Yes.
second order reaction diffusion
Yes.
equation, right?
Absolutely. So if there's any advantage, so this comes from a cooperator who favored this one, but the advantage could be that here we have a more clear phase separation. That might be better than with the diffusion, as the diffusion has always the tendency to spread everything smoothly. That's not so much the case here.
Okay.
That there's such a double well potential behind, and that helps a bit to get things together.
Mm-hmm. Okay. And can I ask another question? You go a little bit further down to your immune therapy, just in the plots that you made. Yeah, right there. So it has this zigzag behavior.
Yeah.
What's the reason for that?
So this therapy is given typically every 3 weeks. So what you see, each zigzag is 3 weeks. That means the patients go to the hospital, they get infusion. Fortunately, the half-life time of this immunotherapy is quite long. That means it's really sufficient to go every 3 weeks, for example. Look here, the time scale is really long. That means what is behind here is just the infusion. Maybe let me go up again. So here it's given when the patients are in hospital. So that's what you see here as a kind of peak, and then it's decaying exponentially. You just do not really see the exponential decay as it's so close together here in this time scale.
It's just like plotted very densely.
Yes. Yes.
The infusion and then the decrease of the compound. So that's why it looks like that.
Yeah, exactly. It's not a numerical issue or so. It's just the infusion comes every 21 days, and yeah, if this one here is 250 days. Yeah, it's clear that you cannot see really nicely the decay.
Okay, so I would just then like to conclude the webinar and thank the two speakers for their kind participation, and also for everybody who joined us today. Thank you very much. And so we conclude the webinar. Thank you once again. Bye.